So for this lecture, you're going to learn something really interesting. You're going to learn how to read a gene, which I think is kind of cool. So we said genes are responsible for carrying the instructions on proteins. And so this is the time where you actually learn how to read that gene and create the protein from the instructions encoded in the gene. So if you remember, we've talked about genes before, and we said that genes are just a portion of the DNA molecule. So you've seen before kind of an image of a chromosome and bands that showed the different areas where genes were located. So genes are just regions along the length of the DNA molecule where there are specific sequences of nitrogenous bases like the A, T, C's, and G's. So it is the sequencing of these nitrogenous bases that encodes how to make a particular type of molecule. And for the most part, we're talking about how to make proteins. So the genes provide the instructions on how to make the protein, but they don't actually do the construction of the protein. They don't assemble the protein at all. They simply are the instructions. So it's just an instruction manual on how to make that protein. You need builders to actually make the protein. So we're going to talk about the two parts that are required for the actual production of a protein. There's what's called transcription and translation. So transcription is when we go from the information on the DNA to the construction of a messenger RNA strand. So transcription means we're transcribing the information from the DNA into a messenger RNA. And then translation is the process by which we're going to take the information on the messenger RNA and actually assemble that protein. So we're really assembling a polypeptide. We're creating a polypeptide chain, and then that polypeptide chain is going to fold into its unique shape, which then becomes the protein. So translation involves the going from one language to another language, right? We're going from the language of an RNA molecule, which is a nucleic acid, to the language of a protein, which is made up of amino acids. So that's why we call it kind of translation, because we're going from one language to another language. So we're going to start off with transcription first. And in this image, you can see that transcription takes place in the nucleus. So step one's in the nucleus. Why in the nucleus? Because that's where the DNA is located. So we don't want the DNA to leave the nucleus because proteins are produced in the cytoplasm, and we need the assistance of a ribosome to help us make proteins. So the protein construction occurs in the cytoplasm, but our instructions on how to make that protein are locked up in the DNA, or locked up in the nucleus of the cell. So we don't want the DNA to leave the nucleus because there's a lot of enzymes in the cytoplasm that can damage the DNA and start degrading it. And that's our instruction manual. We don't want anything to happen to that instruction manual. We need those instructions to keep on building new molecules. So therefore, we need to keep those instructions locked up tight within the nucleus. So instead, what we're going to do is we're going to make a copy of just a portion of the instructions. So if we want to make one particular protein, all we need is the instructions on how to make that protein. We don't need how to make 10 or 20 different proteins along the length of that DNA strand. We just need that one region that tells us how to make that one protein. So we're going to copy that information in the form of a messenger RNA strand. And it's the messenger RNA that leaves the nucleus. The little red string there is our messenger RNA. It moves out through the pores in the nuclear envelope and goes out into the cytoplasm and meets up with the ribosomes for the second half, which is called translation. So, as we said, transcription occurs in the nucleus, and the goal of transcription is to make a copy of that portion of the DNA that we need in order to make a protein. So we want to make what's called a messenger RNA strand. So messenger RNA, or mRNA, are kind of temporary molecules, and they're simply the transcript. So to start off, we are going to use an RNA polymerase enzyme. If you remember, during DNA replication, we talked about DNA polymerase. And DNA polymerase was responsible for making a DNA nucleotide sequence. Here we want to make an RNA molecule, so we're going to use an RNA polymerase. So over on the left-hand side, you see our double helix of our DNA strand, and in pink, it marks the beginning of a gene, and at the other end is a little bit pink area that's called the terminator region, and that's the end of our gene. So we have a kind of a start area and a stop area for our RNA polymerase to work within. So RNA polymerase is going to attach to the area of the DNA that's called the promoter region. So as it says, the promoter marks the beginning of the gene. I always like to think of the promoter as like a sign. So when you're driving down the highway, let's say, and you're trying to get to a certain destination, and you see these road signs, and one of the road signs might say, okay, 5 miles up ahead is the exit that you're looking for. So that road sign is giving you advance notice that your exit is coming up. It's kind of the same thing with the promoter region. So RNA polymerase can be kind of cruising down the length of the DNA molecule. And when it gets to the promoter region, the promoter is saying, hey, 20 nucleotides ahead is the gene of interest. So get ready. You want to start replicating it. So that's our promoter region. It kind of lies outside of the gene, and it allows the RNA polymerase to prepare for the transcription process. So RNA polymerase is going to unzip our DNA molecule. So just like the helicase was doing during DNA replication, RNA polymerase can do this. It can break the bases, break the hydrogen bonds between the nitrogenous bases and expose them so that it can start matching up complementary bases. So in this image below, you see an area where RNA polymerase, our green bubble here is our RNA polymerase, and the DNA molecule has been opened up, all right? And you see this red string falling away. This is the messenger RNA that's being created. But let's look in this bubble here that has been blown up. So this is showing a little bit greater detail of what's happening. So we're copying just one side of the DNA molecule. And we're doing it using our base pair rules. So RNA polymerase will come to a base on the DNA, and it will match up a complementary RNA nucleotide. It'll go to the next base on the DNA, match up a complementary RNA nucleotide, and it'll keep on working along the length of the DNA molecule until it gets to that terminator sequence. So the terminator marks the end of the gene. At that point, the messenger RNA molecule falls away, and the DNA helix wraps back up again. So the DNA is not changed by this process at all. We can open the DNA molecule again and make another messenger RNA from that same site. So the DNA is just our instructions, and we're simply copying it down in the form of an RNA molecule. Now, we said that RNA polymerase matches up RNA nucleotides. So there are a few differences between DNA nucleotides and RNA nucleotides. So with DNA, we knew that the amount of adenine was always equal to the amount of thymine, and the amount of guanine was always equal to the amount of cytosine. It's a little bit different here. The one difference that we have is that thymine is not present in RNA molecules. So thymine is going to be replaced with uracil. So wherever you would normally want to place a thymine, you would substitute it with uracil instead if it's an RNA molecule. So if there's an adenine on one side, then it would be uracil across from it. If there's a uracil on one side, you would place adenine across from it. There's no change with the guanine and the cytosine. So it says if the DNA nitrogenous base sequence is CTATG, what would be the messenger RNA base sequence created from this DNA sequence? So we want to create a messenger RNA sequence. So if there's a cytosine on one side, the RNA polymerase would place a guanine right across from it. If there was adenine on one side, excuse me, I kind of gave it away there. If there was thymine on one side, you would place an adenine across from it. If there's adenine, normally you would want to place a thymine there, but we're making RNA, so instead it's going to be replaced with uracil. Thymine on the DNA would have adenine across from it on the RNA, and guanine on the DNA would have cytosine across from it on the RNA. Right? So hopefully that makes sense. Once again, wherever there is a thymine, you would have adenine across. If there's adenine, you would have uracil across from it. Now we come to a point here where we find that when we make this transcript, this messenger RNA strand, there's actually too much information that has been copied over, and we need to get rid of some of that information. And this is what's called alternative splicing of the messenger RNA strand. So we actually copy too much information. So there are regions that we recognize that are the instructions for the protein that we want to make. And these are what we call exons or expressed regions. And the introns are the areas that we've copied that do not have anything to do with the molecule that we're trying to make. And these are called introns or intervening regions. Sometimes we call them the interlopers. They're coming in and we don't need them. So why does this happen? Why do we have introns? Why are we copying information that's not needed for this molecule? So if you remember, we had a kind of a start area and a stop area for that gene. So this gene contains extraneous information that we don't need to make that particular protein. So if we look at this and this sequence here, and we say, okay, for this, everything that's in black is our exon, and everything in red is our intron. So it's almost like you're watching one of those crime shows, right? And that's everything in black is your crime show. And as you're watching the show, all of a sudden it's interrupted right here by a commercial. Maybe it's Ozempic or something like that, one of those commercials where everybody's dancing around a fountain and you're watching this and going, this makes no sense at all. Where's my crime show? I want that back. And then sure enough, your crime show comes back on. So this is our exon once again. Then another commercial comes on, right? Maybe it's about Snapple or something like that, right? Commercial goes off. Then your crime show comes back on. You finally found out who done it, all right? So in black, these would be our exons, right? We want to watch our crime show. And in red, these would be our commercials. So why does this happen once again? Why do we copy all this and then find out that some of this information's not needed? Well, alternative splicing simply refers to the fact that one gene can contain information on how to make a multitude of different types of proteins. So this kind of saves space, right? If one gene has the instructions on how to make lots of proteins, that's really important. So that the next time we read that gene, maybe these red regions are the exons and the black regions are the introns. And maybe the next time we read it, we splice it differently and create different black regions and red regions. So therefore, just from this sequence here, we can make a multitude of different proteins just in how we view our exons and our introns. So it's all in how you cut it out, right? So once again, the introns we want to get rid of. Those are the interlopers or the intervening regions, and the exons are the express regions, and those are the ones that we want to keep.